let's continue with chapter 6. And I have to post the video lecture video from Friday. There was some technical difficulties with getting it up, but I figured it out today. So, the next part of chapter 6, we went through endergonic and exergonic reactions and how they proceed towards equilibrium. The one part that we left off on was talking about an open and closed system. All right? An open system, what happens is that you never reach an equilibrium. So a reaction will continue to go as long as there's reactants to fulfill that reaction. All right? As long as there's reactants present, that will continue. In a closed system, an equilibrium will be reached, right? So in a closed system, you're going to have a point where there's a medium between the reactants not reacting and the products that are produced, all right, and going back and forth, all right? The point of this being endergonic and exergonic reactions and changes in energy is about to do, or is the ability of a cell to do some type of work, all right? And those reactions that we're talking about, breaking things down, releasing energy, we're utilizing that energy, the cell's utilizing that energy to do some type of work. 3 basic types of work. We have chemical work, all right? We have transport work and mechanical work. Give me an example of mechanical work. What does a cell do that is mechanical work? The beach is that way. What is that? It's a contraction. So muscles. All right, muscle contraction is a mechanical work. Mechanical in terms of moving something. Exocytosis. All right, moving physically, moving something. Molecular motors, right? Myosin and dynein, they react on the cytoskeleton and move vesicles in the cell. That is a mechanical work, right? We're going to physically move something, all right? Transport. Give me an example of transport work. Right? We could put endo and exocytosis in that as well. All right? What other types of transport utilize ATP? Active transport, right? We're going to move things in and out. All right? Active transport. Then the last part is chemical work. All right? And this is probably the most difficult to gauge an example of. Right? But changing chemicals and activating them, moving groups, all right, phosphates or methyl groups or whatever other groups that we talked about in chapter 3, right? Moving them from one chemical to another. That is a chemical work. All right? So the movement or modifying proteins, modifications of DNA, that's all chemical work.
Like cellular respiration.
Yes. Cellular respiration and photosynthesis are examples of chemical work. Right? We'll use cellular respiration as an example. We're going to break down glucose and transfer phosphates to ATP, right, and activate the chemicals. Amy Jo.
Like the making of ATP, I was going to say, I think you might have said it, the making of ATP
is an example of chemical work. All right? To do this type of work, cells manage energy resources by coupling reactions together, what's called energy coupling. All right? This is the use of an exergonic process to drive an endergonic one. So we're going to couple reactions. An exergonic process releases what? Releases energy. All right? When energy is released, then the cell couples the reaction together so that that energy that's released drives another reaction that requires energy. Okay? It's a way of efficiently keeping things going without losing too much energy to the system in terms of heat. All right? And most of these energy coupling in cells is mediated by ATP. All right? And ATP is being the energy currency in cells. the structure of ATP, right? So understanding the structure of ATP will help you understand the energy coupling process, okay? Adenosine triphosphate is a nucleotide, sugar, base, and 3 phosphate groups, okay? It has its role in energy coupling, we'll see that, but it also is used to make RNA, okay? So, adenosine triphosphate, it is a nucleotide, right? The diphosphate or the monophosphate is actually what's utilized to make DNA, but the triphosphate is what's used to make RNA, okay? Here's its structure, right? Here's ribose is the sugar, right? So why is it used to make RNA? That's the sugar that's there. This is a ribonucleotide, okay? The adenine, your nitrogenous base, and your 3 phosphate groups, right? The 3rd phosphate group, the bond between the 2nd and 3rd phosphate groups is a high energy bond, right? So when that is broken, energy is released, and then it ends up being coupled to drive an endergonic process. Okay? And so you see this hydrolysis reaction, H2O goes in, breaks the high energy bond between the 2nd and 3rd phosphate, leaving an inorganic phosphate and releasing energy when that bond is broken. Questions on the structure of ATP. All right? And this is a reversible process. ADP, adenosine diphosphate, can regain that inorganic phosphate to then become ATP again, all right, as the cell regenerates ATP. All right, so the bonds can be broken by hydrolysis. Energy released when that 3rd or that terminal bond is broken. There's also energy that can be broken or released when that 2nd bond, between the 1st and 2nd phosphate, is broken, but it's far less than between the 2nd and 3rd phosphate is broken. All right? The release of energy comes from chemical change to a state of lower free energy, not from the phosphate bonds themselves. So, ATP, okay, has a higher energy state than ADP. Okay? When that bond is broken, ADP has a lower free energy state. Okay? And then that ends up, that release of energy is what's collected by the cell or is transferred from that reaction to the other reaction that requires energy. All right? So how does this happen? So you have 3 types of cellular work that are powered by ATP. All right? Chemical, mechanical transport. Every time you see a reaction that says requires ATP, you have to understand that that is a coupled reaction that's including it. You may see a reaction that occurs, all right, cellular respiration. Overall, the overall reaction for cellular respiration is C6H12O6 plus O2, right? Going to break down and produce CO2 plus H2O plus ATP, right? So what's happening, there's no products, or you also have to include ADP plus inorganic phosphate in there because this is what's happening. ADP plus your inorganic phosphate. Oh, something just retweeted. Leads to ATP. All right, that's the background equation that's occurring. That's happening, how the cell stores ATP. Okay? So understand when ATP is utilized, this reaction is ongoing because this actually goes both ways. All right? So anytime you see, say, a chemical reaction, whatever that requires energy to be put in, or ATP, this reaction that's your coupling reaction. All right? So the hydrolysis of ATP plus H2O, right, can be used to drive an endergonic reaction. So here's what it looks like. So glutamic acid plus ammonia, all right, converts to glutamine by itself, all right, and that's has a certain energy, that state, right, 3.4 kcals per mole, all right. What it actually has, this is the basic outline, but what's actually ongoing is that glutamic acid is along with the hydrolysis of ATP, is now changing the state of that glutamic acid. The phosphate has been transferred from ATP to the glutamic acid. Now it's phosphorylated intermediate. That phosphorylated intermediate has a higher energy state, okay, and is reactive. It will react with the ammonia, okay, to form this product. All right, so this intermediate is important. There's an intermediate step. Although you see this reaction up top, right? You see this reaction occur, or you may actually see, plus ATP. You have to understand that this is the intermediate that's ongoing. Okay? So to see it with the conversion process of glutamic acid, right, ammonia plus the ATP, here's the free energy change for the couple reaction. All right? So by hydrolyzing ATP, you have a negative 7.3 kcals per mole, right? What does a negative free energy mean? That's going to happen how? Spontaneously. Release of energy is going to occur. That energy is transferred in the form of the phosphate to the glutamic acid, right, which makes it reactive. And now that proceeds spontaneously to this. Okay? So you end up with an overall, you have the negative 7.3 and the 3.4. You end up with a negative 3.9 kcals. It's still a negative. That means this reaction, coupled with the hydrolysis of ATP, occurs spontaneously. It is really warm in here. It's okay. Questions on coupling? So there's really two reactions that are ongoing here. ATP is being hydrolyzed into ADP and inorganic phosphate, and that hydrolysis is driving the transfer of glutamic acid to glutamine. All right, so glutamic acid plus ammonia to glutamine. Right, an endergonic reaction is driven by an exergonic reaction, the hydrolysis of ATP. Okay, so this typically happens by phosphorylation, and you'll find this in cells. As you move up in courses, we talk about cell signaling, or when we talk about cellular respiration, you'll see the transfer of the phosphate group from ATP to intermediates. All right? That is how ATP drives this process. Those intermediates have a higher free energy, which then allows them, they become reactive, and then the process proceeds. Okay? This is your phosphorylated intermediate. Typically, there's a phosphorylated intermediate. All right, so the first step in cellular respiration is glycolysis. Okay? In order for glycolysis to occur, glucose goes in, and ATP is utilized to split glucose into 2,3 carbon sugars. And it does that by moving 2 phosphates to the glucose, producing glucose 6-phosphate. Glucose 6-phosphate is more reactive than glucose, and then it splits in half. All right, and you will go over all those steps sometime next week. Okay? But the point being is, in order for that to occur, the phosphorylated intermediate is utilized. All right? So ATP hydrolysis can lead to the change in the shape of a protein, or the reactivity of a molecule, and can also affect the ability to bind and react with other molecules, right? By adding phosphates or changing phosphates, you can alter the shape of proteins or their activity of molecules in general. Whew. Okay, so here's a couple of examples. Transport protein, right? That protein itself is an intermediate. Okay, when ATP is hydrolyzed, that phosphate is transferred to the protein within the membrane, causes a shape change. That shape change then allows whatever solute to bind, right? Whatever molecule is being transported to bind, then that causes a shape change and it's released, and then inorganic phosphates released. All right, so the change of the shape in terms of protein just by adding or removing a phosphate is important. All right, the other mechanical work, right, the movement of motor proteins. All right, so here's a vesicle attached to dynein, your motor protein. ATP is bound, it's hydrolyzed, changes in shape. It allows the molecular motor to walk along the cytoskeleton, right? Because every ATP that's hydrolyzed causes a stepwise shape change and allows it to rebind to the next or the subsequent binding spot, right? Similar events occur in muscle contraction. Actin and myosin, right? Hydrolysis of ATP allows actin and myosin to bind, right? And then also ATP has to be hydrolyzed to break that actin-myosin association. Now, I said that ATP can be regenerated, all right? So the overall reaction is adenosine diphosphate, or, plus the inorganic phosphate leads to ATP, okay? Where does this come from? Again, this is a coupled reaction. Something happens in the cell, something's broken down, energy is released, and it's stored. That energy is stored in the form of ATP. All right, energy is released from glucose, from a catabolic reaction, and then it's stored in the form of ATP. All right, so this ATP cycle is what they call a revolving door. It's going to transfer from ADP to ADP and then back. All right, so this ends up, we can just do a cyclic type of thing that occurs. All right, so you go from catabolic to anabolic, this one.
Yeah.
Okay, so what's happening here? What's happening in these exergonic reactions?
Is it so the surroundings are becoming? Is that what it's talking about?
So, I'll give you an example and we'll see if we can work through this. So, glucose, C6H12O6, right? It's going to be broken down into CO2 plus H2O, right? In the presence of oxygen. Okay? And then that is an exergonic reaction. This part is. Okay? Meaning that the breakdown of glucose, breaking it from its ring structure into smaller parts, into CO2. Every time you break those chemical bonds, there's going to be a release of energy. Okay? To the surroundings. And then coupled to that, next to that, the cell is going to store that energy that's released by transferring ADP plus your inorganic phosphate into ATP. Okay? That's building this. What?
No, that's not the physics stuff.
Okay?
Okay.
Does that clear your
Yeah.
question? And then on the opposite side of that, you're going to break down ATP to ADP to help run those endergonic reactions that are going to require. So building amino acid 1 plus amino acid 2, right, to a protein that requires energy. And that's what ATP is utilized for. Okay? Other questions? Questions on coupling reactions between ATP and the hydrolysis of ATP and endergonic reactions? Yes.
So, like, I think I sort of understand what you're saying with, like, energy coupling, like, so if, like, one part of the cell needs ATP for something, then, like, like ADP and the free like phosphate will somehow like get detached from that and get used for something else in the cell.
Mhm.
It's like the way it works.
Basically, the cell will always be producing, ideally, the cell will always be producing ATP, breaking down glucose as it's necessary, or other proteins, fats that are present within the cell to produce ATP. Okay? So there's always, anytime, if there's ever not enough ATP, the cell dies. All right? You all die from various reasons. The last one being, the most ultimate reason being, you run out of ATP. Okay? So, it is always a net gain. The cell is always doing something to store and have ATP present, so that within the cell, it can be utilized to do the things it needs to do, whether it be replication of DNA, movement of proteins, production of proteins, etc. All right? And then once it's utilized, the ADP and inorganic phosphate are available to reproduce the ATP itself. Right? That's overall cellular as it might be. Okay? Any other questions? Okay. The next part, Danielle, sorry.
Sorry, so we were a little confused, when it's exergonic or endergonic, is it related to the surroundings, like the surroundings is becoming, like the little diagram?
Mm-hmm.
Because it says energy out, endergonic, but isn't that exergonic?
If energy's being released, it's exergonic. Okay? And tends to be a spontaneous process. Okay? So you're going from a high energy state to a lower energy state.
No, I know that, but like the little diagram, it says energy out and then under it, endergonic.
What diagram?
On the, what's just happening?
So enzymes catalyze reactions by lowering the activation energy. They do not affect the change in free energy from products to reactants. They don't change the energy state of the reactants or where they're going. Okay? Doesn't change the product or the reactant's free energy state. It just changes how much is necessary, how much energy has to be taken from the environment necessary to get that reaction to occur. All right? So they actually speed up reactions that would eventually happen without utilizing so much energy. All right, so reactants and products are still the same, and then that's work, but it doesn't work. This is your overall. All right, the state of the reactants is still and the products are still the same. Okay. What ends up occurring is that the lowered activation energy is present and the reaction occurs at a faster rate. Questions on enzymes and what they do in terms of their catalyzing effect, characteristics. How do enzymes work? Gave you an example of how sucrase works on sucrose. Binds the substrate and causes a tension on that bond. Tension on the bond allows, actually physically lowers the activation energy and allows the reaction to occur. All right? So substrate enzyme are shape identification between the two. All right? They're dependent upon shape identity. All right? Sucrase works on sucrose. It does not work on lactose. Okay? How many of you are lactose intolerant? Okay? Which means what? You lack or have a lactase. You have a inhibited or lack of an enzyme. Okay? I am alcohol intolerant. No joke. Asians People of Asian descent your body has 3 alcohol dehydrogenase enzymes. Okay? 3. That means in your blood, alcohol is broken down from ethanol to the other products that it ends up with. Right? You have 3 of them, 3 copies. If one of those is mutated, right, which people of Asian descent tend to have that mutation, you cannot process alcohol as fast as others. Okay? So, your 2 enzymes work just fine, right? 1 doesn't. You end up with characteristic side effects, blushing of the face. Some of you might have seen this in terms of sulfites, in terms of wine. Your parents drink wine, one of them gets red, right? Yes, this occurs all the time. All right? So, it is not just lactose. There are other enzymes that can be mutated that you are intolerant for. And it has everything to do with that 3rd enzyme doesn't recognize the ethanol, the structure, its substrate. All right? So enzyme binds to its substrate, forming an enzyme substrate complex. Right? And your active site is the site where the point, how the enzyme works, where is actually occurring. All right? So where the substrate binds and it occurs, does its magic. All right? But specificity, enzyme substrate specificity is important. Otherwise, the enzyme doesn't work. And you did this in lab by, ideally, denaturing the enzyme through heating, changing the shape of the enzyme, and now preventing it from binding to its substrate. There's no more enzyme substrate recognition that occurs. Okay? So enzymes change shape due to chemical interactions with the substrate. There's this idea of what's called induced fit. All right? It is not lock and key fit like it used to be. There's actually induced fit. Okay? Enzyme substrate brings chemical groups that fit together or will react into positions that enhance their ability to catalyze the reaction. All right? So how an enzyme binds to a substrate now induces, there might be a little shape change of the enzyme so that the substrate is bound and then can bring chemical groups together that will allow this to occur. That will have the lower the activation energy and help speed up the reaction. Okay, so here's a substrate, enzyme, substrate binds to the active site, causes a change in the shape of the enzyme. Right, so there's a change in the shape. This is your enzyme substrate complex. What occurs then, right? There's a change in the substrate, whether it be the breaking of sucrose and glucose, or sucrose into glucose and fructose. And once that occurs, now it doesn't bind it as well. Another shape change. The enzyme changes back to its original shape, releasing glucose and fructose, and then binding another substrate. All right? What has not occurred in this reaction, the enzyme has not been altered. All right? The enzyme can be reutilized, all right, and recycled. Within the active site, how can this occur? All right, 4 ways. So it can orient the substrates correctly. So you put them in the right position so that they have one chemical group that's going to react close to another chemical group that's going to react. Put them close to one another, the reaction occurs on its own. All right? Otherwise, they could randomly end up together, but you're going to quicken the process by getting them close to one another. All right? An enzyme can put strain on the substrate bonds, such as in the case of sucrose and sucrase. All right? It can provide a favorable microenvironment by shielding the substrate from the outside. You can increase the pH or the temperature or alter the presence of hydrogen bonds, and then will alter the substrate itself. Or it can actually covalently bond to the substrate, providing an intermediate and providing a higher reactive state of the substrate itself. All right, so here's your overall picture. All right, in this case, what type of reactions occurring? You have two substrates come in, right? They come close to one another, provide the right fit. Substrate's held in the active site for a little bit. All right, by weak interactions, hydrogen bonds, van der Waals, forces, whatever it might be, you've now produced the enzyme substrate complex. The presence of the enzyme or with the substrates causes a change, right? They're converted to products. Once they're converted to products, they no longer bind to the active site as well as the reactants, right? This allows their enzyme to go back to its original shape, releasing the products from the active site. And the enzyme then is available to bind another set of substrates. Questions on enzymes and enzyme substrate recognition? All right, we'll stop there.